So it's my pleasure to introduce tonight's speaker, Jeff Lichtman, the Jeremy R. Knowles Professor of Molecular and Cellular Biology, and the Ramon E. Cajal, I may have said that wrong, Ramon E. Cajal, Professor of Arts and Sciences at Harvard University, where he is also a member of the Center for Brain Research. He earned an AB from Baldwin and an MD and PhD from Washington University, where he was a professor before taking up professorship at Harvard. His research seeks to understand how circuits in the brain are altered by experiences, especially in early life, particularly the dramatic rewiring that takes place in early postnatal development when a tremendous expansion of learning takes place. As part of this work, he developed methods for visualizing neural connections and how they change over time, perhaps most notably the rainbow method. He has also been developing electron microscopy methods to map wiring diagrams of developing and adult brains with exquisite detail and completeness, and with the very particular aim of understanding how information is stored in neural connection networks. His talk tonight is entitled Connectomics, Mapping the Brain's Wiring Diagram. Jeff, the podium is yours.
Well, thank you very much. It is a great pleasure to be here and to join this very old society. I am a neurobiologist. I'm assuming most of you are not neurobiologists, and I may occasionally lapse into neurobiologies. And if you are completely confused by what I say, if you just make a funny face at me, I'll try to restate things in a slightly clearer way if I can. Biology generally is interested in living things, but one of the oldest themes in biology is the relation between the physical structure of living things and their function, structure, function. The state of the art in biology right now is a field called structural biology, which sounds like a very general field, but it's actually quite a narrow field. And it's a field that focuses on the atomic structure of molecules by looking at exactly where the atoms are in a molecule. People who study molecules at this level of resolution try to understand how the molecule does what it does, if it's an enzyme or some kind of factor that turns on or turns off DNA, for example, or binds a particular ligand. Structural biology is the way people study this. Now, that's the state of the art, but there probably was a time in the history of biology when structural biology, it wasn't called that, dealt with much bigger things than molecules. Probably there was a time when humans first began dissecting open the animals they were eating, or perhaps cadavers of human beings, and they noticed these big aggregations of tissue in various parts of the abdomen, perhaps in the head as well, that looked sort of the same from one animal to another. Even though a zebra and a lion were completely different on the outside, you could recognize the liver, you could recognize the kidneys, because they look relatively similar, even in a human and these other animals. And for most of these organs, it was clear they were tissue, but it wasn't clear what they did. In fact, the only organ that really very early became straightforwardly understood was probably the heart, because with your naked eye, you could see this thing contracting if the animal was not completely dead when you opened it up, and understand that this was a pumping organ. But for the other organs, although you could recognize them from animal to animal, you had very little idea what they were able to do until the discovery of a very fundamental tool. Of course, was the microscope, which allowed humans to discover for the first time that these organs are made up of cells, and the cells are put together in motifs that are the same for one organ to another example of the same organ. So for example, if we think of the kidney, there is this motif known as the nephron, which is just a little tube that's like a filter. And this filter ultimately explains what the kidney does. In fact, the kidney is quite a boring organ because once you understand what one nephron does, you basically understand the kidney entirely because it's just made up of hundreds of thousands of these nephrons. And in the liver, there's something called the portal triad, and there's the alveoli and the bronchioles in the lung, which is just a motif in each of these organs that does the function of the organ. And once you understand that, you have a good idea what the organ does. In the lung, it's gas exchange. In the kidney, it's filtering. Not only do you understand what it does, but you also understand what's wrong with these organs and diseases. And the field of pathology is a field that studies these cellular motifs and how they're deranged in various diseases. And this has been a profound insight in the medical model of disease. That is, we not only have abnormalities due to disease, but we actually have physical underpinning for that. This has worked out really, really wonderfully for all organs of the body except for one organ. And not surprisingly, that organ is the most important of all organs, and it is the brain, where we don't have a good idea about these cellular motifs, and even worse, we have lots of diseases where there's no doubt something is wrong, but we have no idea what physically is abnormal, and that's partly because we don't even know what normal looks like. We don't have good neuropathology for most diseases that are called psychiatric diseases, most learning disorders in children. These are profound diseases, a serious drawback for our understanding. I'd like to, I'm giving you this long introduction because I'm trying to justify something that will seem patently insane to do, which is to try to turn the brain into an organ just like all the other organs, made up of cells, and we will understand someday how these cells are put together. So I want to begin by trying to justify for you why is it so hard to do this in the brain compared to these other organs. And so why is the brain so hard to understand? Compared to other organ systems of the body, the relationship between the brain's physical structure and its function is much more complicated. And there are three reasons for this complication. And the first relates to these cellular motifs. If you think about the cellular motifs for a kidney, according to Wikipedia, there are 5 types of cells in the liver and maybe 10 types of cells in the kidney. And those cells are put together into these motifs. Does anyone here have an idea how many types of different cells there are in the brain? 50? Yeah. So 50 is a good number, but that's the retina. So here is from 2001, Dick Maslin's description of all the different kinds of cells in the retina, which is one little part of the nervous system. And what he's done is he's traced out the shapes of these different kinds of cells, starting from the photoreceptors that respond to light. There's a red, a green, and a blue photoreceptor. And then there are these horizontal cells, bipolar cells, amacrine cells, and ganglion cells are the cells that leave the retina and send visual information into the thalamus, into the brain. And he, in 2001, could already see clearly, distinctly, 50 types. This is actually now a state-of-the-art research question right now. Neuroscientists are writing grants to figure out exactly how big this number really is, and it's certainly bigger than 50. Some people think it'll be 100. Some people think it'll be much more than that. Some argue that perhaps there aren't even types, but a continuum. There are thousands and thousands of different types. We don't know. Someday we will know. The retina is almost the best place you could possibly ask this question because it's a tiled situation. It's like a camera faceplate. Each part of the retina has to cover the same kind of information. So if you see a cell type and you see it over and over again, you know you've caught a cell type. Someday we're going to get that number for the retina. But guess what? In the cerebral cortex, there's a completely different menagerie of cells that have nothing to do with these cells. And in the cerebellum, another set of cells. And in the brainstem, and in the thalamus, and in the spinal cord, every part of the brain has its own unique, massively diverse set of cells. There's nothing like this in the rest of the body. All the cell types in the rest of the body combined is far less than the number of different kinds of cells in the nervous system. This is a big problem, and that's the first problem. So far greater structural diversity. And the second is functional diversity. You know, brain does a lot more than the kidney does, and I'll have a lot more to say about that. There's a second problem about the brain, and maybe the easiest way to make this case for this fact that the brain is organized at many size scales, which may not seem like a big problem. If you want to do a good deed, you may donate a kidney to someone else, and that will impact your kidney function, but almost not at all, because there's a lot of excess capacity in our kidneys. You can also survive with half of your lung tissue. You can't run a marathon, but you can survive. You can lose a substantial portion of your liver and be fine. But no matter how I cut your brain and take half of it out, you will notice. It'll be a big, big problem. And that's because there is no extra capacity in that sense. Every part of the brain has its own particular function that's distinct from other functions in other parts. So we look at the brain as sort of a simple cartoon from the front of the brain to the back of the brain in cross-section. If you're planning about what you're going to do later tonight or tomorrow, that'll be related to your frontal cortex. If you're watching this lecture, you are driving cells in the back of the brain, the occipital lobe, for vision. When you get up, if you don't fall over, that you can thank your cerebellum for that. If you're breathing, you can think how important your brainstem is, and to walk, you need your spinal cord. Each part of the brain has its own particular functions. That kind of division of labor does not take place in most other organs. I want to now zoom in progressively higher resolution to smaller and smaller parts of the brain to show this kind of division of labor is not just this gross division of labor, but even down to the finest level. So let's just look at this little region of the cerebral cortex. And here, I'm sure you've all heard the term gray matter and white matter. I've made this very confusing by making the white matter pink and the gray matter gray. The gray matter is where the nerve cells are in the cortex, in this rind around the outside. And the white matter are where the cables, which are axons of nerve cells, leave nerve cells from one part of the brain and go to another part in these white matter tracts, as they're called. So there's a division of labor there that's complicated. That is, it's not a uniform tissue. It's got a white and a gray area. And then if we zoom up on the gray matter and look at what the cells that are located there, the nerve cells known as neurons, you see that these cells are just not like cells anywhere else in the body. And here's an example of one of these cells. It's called a pyramidal neuron because the cell body is sort of in the shape of a pyramid. It's kind of triangular. And this cell, in distinction to cells elsewhere in the body, have this massive number of little things sticking out of them. These are dendrites. They're the antennas of this cell. And this cell also has one wire coming out called the axon, which is the cell's output. It's a little hard to understand what this is about without appreciating that this cell is sort of like, it's got I don't know whether Twitter or Facebook is the right analogy because I don't use either of them. I'm showing one of the limitations of my brain, and I'll come back to that idea many times here, but this is a cell that receives information from many friends. And because it wants to get information from many friends, it has these wires stuck out all over the place that are places where other nerve cells talk to it with synaptic connections. So this cell is receiving input from thousands of other cells, And then this cell has many followers. That is, its axon, it sends information to thousands of other cells, and this axon could cross to the other hemisphere or go down the spinal cord. So this cell itself is drawing information from a huge number of other cells and sending information to a huge number of other cells. This is a network very similar maybe to an internet network of human beings, your friends. And I don't think that's a coincidence. I think, of course, when your friends talk to you, ultimately they're activating the dendrites of cells that respond to sound. That information is getting into your nervous system and propagating throughout your nervous system, and if it ends up down in your fingers and makes you type something, you're going to be sending that information ultimately into the brains of other people. So basically, not only is one brain a vast network of interconnected wires, but we are now part of this vast, gigantic brain interconnecting all of us. And I think biologically it seems very different, but fundamentally it's just a different form of neurotransmission when you go from out of one brain and into another. But I want to just go on a little bit more. We're down to a millimeter size scale here. If we zoom up on the dendrites of this cell, you see another division of labor. The dendrites are the receptive side, but they themselves are complicated. If we zoom up on that little area, the dendrites of these cells have these little thorns coming out, known as dendritic spines, and this is where the contacts from axons of other cells make synapses onto this cell. The contacts onto the spines try to get the cell to wake up. They excite this cell. There are other synapses and connections made on the shafts in between the spines, and those are from nerve cells that are trying to shut the cell up. They're called inhibitory neurons, and most of your brain, most of the time, is being inhibited. It's a good thing, otherwise you'd be spontaneously blurting things out all the time while I'm talking. So inhibition is important. It keeps us quiet, and only occasionally does something drive a cell enough to get information to flow. And I'll explain that more a little bit. We zoom up on one of these spines and go another order or two of magnitude. I now have to shift to electron microscopy, to very high resolution images, to give you a sense of what a dendritic spine looks like. So here's a dendrite, and coming off of it is this narrow piece that ends in a slight swelling, and that's a dendritic spine. And that spine is up against a cross-section here. This is an electron micrograph of an axon that's filled with these little circles, which are round spheres filled with neurotransmitter. And that is the chemical that is released by this nerve from the axon terminal across the synaptic junction to send information from the axon to this dendrite. You see cells have thousands of these spines, and this is what one of them looks like. So let's just pause for a moment and see why this is a problem, this multi-scale information. If you want to understand a brain at this level and use the highest resolution techniques that allow you to see different parts of the brain that are active at different times, this is a technique called functional magnetic resonance imaging, and it has a resolution of about a cubic millimeter. That is, a human brain might have 1 million of these voxels, and I'm sure you've seen pictures of brains where they light up a part of the brain associated with a particular function. That is the resolution, the state-of-the-art resolution of imaging whole brains in human beings. If you image brains at the resolution to see synaptic vesicles, which is where the information is passing from one cell to another, you're doing that at a resolution of 100 cubic nanometers, and that means one voxel of this image is 1 trillion times smaller than one voxel of that image. Put another way, 1 trillion of these fit in each one of those. And if you want to understand a brain, you've got to understand it at this level, this level, this level, this level, and that level. And no matter how you look at it, 1 trillion is a big number. So that's the second reason it's so hard to understand the brain. There's a third reason, and I think this is maybe the most interesting, and it separates the brain from all the other organs as well, and that the brain, like other organs, came into existence in your development through a book of instructions known as your genome, your genes. Your genes build your brain, and that structure generates a brain that has function. And biologists love this kind of linearity because it means if you have a mutation in a gene, you might have a change in the structure and ultimately a functional consequence. And for most of the organs of the body, this is the way things work. It is this kind of linear stream of information from genetic information that you inherit from your parents that build the structures of the body and then generate the function. But with the human brain, there's more to it, and the brains of all mammals, there's more to it, in the sense that there's a lot of functions we have that could not have come from our genes. I'll give you a simple example. Many of you probably can ride a bicycle. Is that true? Is there anyone here who did not learn to ride a bicycle as a child and has tried? And did you learn to ride as an adult?
Yes.
How was it?
Hard.
Really hard. Much harder probably than for a child to learn. Do you now ride regularly?
Yes.
Yeah. And so you've mastered it. How old were you when you learned?
18.
18. Has anyone tried to learn older than 18? And?
31. I found it pretty easy.
Yeah, oh, I'm impressed. And you never rode as a child?
Right.
But once you learned, do you have to practice to keep up with it, or it's now? Yeah. Once it's in your brain, it's fine. You've learned it, and now you have this function. But is that because you have a gene for bicycle riding? Bicycling? No. It must have been somehow that the use of the brain, either as an adult or most often as a child, through practice and use of the brain, you generate a function that once you've learned it, you don't forget it. It just sits there forever. And so it must be stably maintained in the structure. So this arrow is actually going in the wrong direction. It means somehow the use of the brain has generated a physical change in the brain that then generates a new function. And these loops, there are loops all through biology. I just want to tell you a little secret if you're not a biologist. Biologists hate loops. We don't like loops because there's no cause and effect. It's very hard to get a handle on this. There's non-linearities with loops. So we prefer that they didn't exist. But this is how everything that humans do. If you think of your behavioral repertoire today, how many of the things you did you learn? You learned virtually everything you did today except sneeze, breathe, and a few other bodily functions. Most everything else you did, from buttoning your shirt to walking, humans had to learn how to do. This is not true for many other kinds of organisms where that information comes from the genes. Humans are kind of dependent on learning for things that other animals could do naturally. You might wonder, why would we go to the trouble of learning to do these things? Why does it take a human a year to walk? Other animals walk easily. Why does it take humans a year to figure out how to walk? And 15 more years after that before you get your driver's license. And 2 more years after that before you leave the nest when you're 18. I mean, this is crazy. And I think maybe the magic of being a human being is that a lot of the structure of our brain did not come this way. It came from practice and learning about the world. And in my field, you don't become a master until you're a full professor. And you know how long it takes to do that? You go to college. You leave the nest at 18, then you spend 4 years at college, and then you have to go to graduate school. And then you do that for 6 years. And then you have to do a postdoctoral area for, maybe 3, 4, 5 years. And finally, you get a job as an assistant professor. And then you do that for 8 or 9 years. Then what? Then you're promoted to associate professor. Then you do that for another 5 or 6 years. And then finally, someone says, yep, you now know enough to be called a master. By then you have gray hair, you don't give a shit anymore to be perfectly blunt about this. But this is the magic of being a human being. That is, you end up with a massive amount of information that didn't come in this way, but came in through this loop. This loop is fundamental because memory, all those memories we have of everything we know how to do, every episode of our lives, came in through the experience of the world that has changed us in a stable way so we cannot forget. They're indelible, basically, indelible memories, and that is structure. So what is that structure is a big question. Now, this idea of turning memories into something physical is the idea of an engram. Richard Semon in the 1890s coined this term, engram, as the physical instantiation of a memory. So this process, if it had a name, might be called the engrammatization algorithm. That's just, of course, a completely made-up word, but that is what I am interested in. How does experience turn into physical structure? Now, to talk a little bit about this, I just want to remind you, and for some of you, you may not know this, what the structure of the brain is, or at least what we understand it to be. This is based on work that was done in the 1870s and 80s by two extremely talented neuroscientists. One of them was an Italian physician whose name was Golgi, Camille Golgi, and when he was 30 years old, playing around with the materials that allow for photographic emulsions. In his kitchen, he mixed together two salts, silver nitrate and potassium dichromate, and found a very interesting result. He found an extremely inefficient stain for the brain. Had it been a really efficient stain, that is, had it stained everything, it would have been useless. What made it so special is that it was very finicky and only a small number of cells labeled, but the cells that labeled happened to label in their entirety. And it's not entirely clear what has happened, how this works, but the thought is that the silver dichromate crystal is very unlikely to form, but once it forms, it is the nidus for further crystallization, and the membranes of cells, which don't allow water and salts to cross, sort of block the precipitate, which is this dark brown color, from escaping one cell. So you have a very low likelihood event, but when it happens, you can see an entire neuron completely surrounded by unlabeled cells. And this provided another scientist. Camille Golgi was the person who invented the technique, but the person who really used it was a remarkable genius, Santiago Ramon y Cajal, a Spaniard. These two guys hated each other. In fact, they only met at the Nobel Prize they shared, and Golgi at that Nobel lecture just pooped on Cajal the whole lecture. His lecture is very embarrassing because he just couldn't see in his own material what Cajal saw when he began looking at tissue like this. So Cajal made these really beautiful drawings. His drawings were wonderful because they got the essence of nerve cells in their entirety, and he saw something that was so fundamental that it has stood the test of time, and it is largely dominates the way modern neurobiologists think about the brain. And I'll give you a sense of this. Here's a picture of Cajal in his later years. He was, by the way, a photographer largely of himself. There are about 10,000 pictures that he's taken of himself. He has usually a little bulb that he squeezes, so in almost all pictures of himself, one of his hands is clenched because he's taking his own picture. He took his family occasionally, but that was usually, I think, only getting it set up so he could take a picture of himself. He thought highly of himself, but he should have. He was really a remarkable person. So what was the insight he saw? So by looking at this Golgi stained material, Cajal drew pictures like this. Now, the paper has yellowed, but he used whiteout. This is not snow, so the whiteout has stood the test of time, and that's a distraction here. The thing I'd like you to look at is these arrows. This was Cajal's fanciful idea of how information would flow through the brain. He said that the axons of nerve cells end up in a bunch of branches that overlap the dendrites of other cells, and information from that axon transfers over to the dendrites, gets to the cell body of the target cell, and then that target cell sends that information out its own axon, and that axon touches the dendrites of other cells. That information flows down the dendrites towards the cell body. Here's another axon from another source, also touching this cell. And together, these two inputs onto this pyramidal neuron send their information and collaborate in a way to send information out that cell, out its axon. That axon touches the dendrite of another cell. That information flows to the cell body and out its axon. That axon goes on and does the same through this cell as well. And here, for a piece of cerebral cortex, was Cajal's idea of how information might flow through circuits. This idea is very, very compelling. In fact, I'm sure all of you can understand it. And I'm talking about the most complicated tissue on the planet. And here was a simplifying idea that, to a first approximation, is not incorrect. This is true that axons send information out, the information is transferred at dendrites, and these cells then send information further out. This idea has been so dominant, modern neurobiologists, if you ask them to draw a circuit of anything, they basically use this Cajalian shorthand. I'll give you an example of that here. This is a sparse worldview. I just want to remind you, it's not based on seeing everything. It's based on seeing a very, very small subset of what's actually in a tissue like this, maybe 1%. But that sparse worldview has dominated the way we think. So here, for example, from 2002, Rodolfo Llenas, who is one of the masters of understanding how the cortex works, has drawn a diagram of the way cells interact with pyramidal cells in the cortex. And you can see shades of Cajal in this modern drawing. The only real advance, I would say, in this drawing that isn't found here is that we now know that there are this subset of nerve cells that are trying to inhibit information flow, as well as excitatory connections. There are also inhibitory connections. And so these inhibitory cells are put in here, whereas Cajal had no way, just by looking at the Golgi stain, to appreciate that all of this is not kind of trying to push information forward. Some of these cells are trying to slow it down. So this is where modern neurobiology is. But it is important to realize that this is a worldview based on the Golgi stain. Cajal understood that a lot of information is ignored in diagrams like this. I'll give you an example. He changed the staining parameters and looked at this region of a brain and just showed what was present here if you allowed lots of cells to be labeled. And here's his drawing of the superficial layers of cerebral cortex. It's just a little slab of brain, lots of cells, and look for the arrows, and you won't find them, because he was a genius, not an idiot. He knew it was impossible to kind of put arrows on something as complicated as this. Now maybe if each of these cells was a different color, or you had enough resolution to see exactly which connection was which, it wasn't all superimposed, maybe one could make some progress. But this is kind of how we leave the brain now. We just sort of generally say, the brain is very complicated, there's lots of cells, they're interconnected, but not seriously is anyone going to try to map out all these wires. But this is exactly the same way people thought about genomics. When it became clear there were lots of genes, someone at some point finally said, we should map out every one of those genes. We should do a genome, we should do genomics, and mapping out all the genes has turned out to be a really powerful way to see the underpinnings of that instruction book that builds organs and underlies diseases and health. Could we do the same for the nervous system? Could we map out the entire wiring diagram of a nervous system? If we did, it wouldn't be genomics, it would be connectomics, and I know this is a new term for most of you, so I have brought the dictionary definition of connectomics here. It's pronounced connectomics because I assume most of you don't know this term, is that right? Connectomics or connectomics. It's a plural noun, but singular in construction. This is from Merriam-Webster's Unabridged Dictionary in 2019. It's a branch of biotechnology. It's biotechnology. It's not biology, it's technology, concerned with applying the techniques of computer-assisted image acquisition and analysis to the structural mapping of sets of neural circuits or to the complete nervous system of selected organisms using high-speed methods. If you don't go fast, you'll never finish. I'll make that clear in a moment. With organizing the results in databases. And by databases, I don't mean Excel spreadsheets. I mean image databases, things that we don't have ready. It's hard to imagine what an image database of the brain would look like. I'll give you some hints a little bit later. And with applications of the data, as in neurology or psychiatry, that is, there are probably diseases of the wiring diagram, something I would call connectopathy. Maybe these connectopathies will be in this dictionary as well. And fundamental neuroscience questions like, where is bicycle riding? What does it look like? What does your grandmother's memory look like in your brain? Those kinds of questions can't be answered, I don't think, until we get serious with mapping out the wiring diagram at the level at which it occurs, which is down at the level of every axon, every dendrite, every synapse. And the word connectome, might be like the word genome, a full wiring diagram. Our initial attempt to figure out how to do this was to take advantage of the Golgi stain, which was a wonderful technique, except that because it only labeled a small subset of cells, you could never see the whole wiring diagram. And as I mentioned, if the Golgi stain stained everything brown, the brain would just be a big brown block. So what we thought is maybe we could make a Golgi stain equivalent, but make each cell a different color. Not all brown, but every cell a different color. And we took advantage of the fact that there's been a revolution in cell biology in the fact that humans have figured out how to get jellyfish genes inside mammals. And these are genes for fluorescent proteins from jellyfish and from corals. There's a green fluorescent protein, and there's corals that glow red when you shine green light on them, and ones that glow blue when you shine ultraviolet light on them. So they're red, green, and blue fluorescent proteins. And in addition, as we've understood the green fluorescent protein better, that gene has been mutated to generate this extraordinary range of colored fluorescent proteins. So there's lots of colors out there, but actually to generate a different color in each nerve cell, you don't need hundreds of different pigments. You really only need 3 pigments. You only need a green fluorescent protein, a red fluorescent protein, and a blue fluorescent protein. The reason that's all you need is that's all we can do with our eyes. We only have 3 photoreceptors. All the colors you see in the world are just mixtures of the amount of red, green, and blue. So what we did, and Jean Levet, who was a postdoc at the time, and now he has his own lab in Paris, and Josh Sains, a colleague of mine, went about trying to figure out how to randomize the amount of expression of 3 different genes in each cell in the nervous system. And I won't go through the molecular shenanigans we did. It's just ways of just sort of getting different amounts of these 3 genes expressed in individual cells. But we made mice engineered that have genes for red, green, and blue fluorescent protein, and they are expressed in the nervous system in a way that each cell has a stochastic amount of these 3 proteins. And so each cell has its own RGB signature, and you get all the colors you get out of color television just by mixing different brightnesses of the red, green, and blue pixels in different places. So this is a cross-section of a little piece of brainstem showing these axons that are running in this area, and you see that there's lots of color. These are axons that are running along the path, and they're superimposed on top of each other, so you see a whitish glow because we're seeing many colors on top of each other. Here's another picture. There's just lots of color. And if we zoom up on this area here, you see that these colors are easily distinguished one from the other. In fact, if you focus up and down on data like this, And that's what we're doing here with something called a confocal microscope. You can follow these wires as you're moving through this little slab of brain. So this is an approach. And it's not that this stuff is really moving. I want to make this clear. This is not a time-lapse image. This is just a space-lapse image where we're focusing on progressively deeper part of this little piece of brainbow mouse brain. And because, let's say, these wires here are running diagonally through the data set, they seem to appear and disappear. They are always there, but we're only looking at that depth at one moment in time. Is that kind of clear? We're going to see a lot of pictures like this. And here's the cerebellum. The colors now are fantastic. We've made new versions of these that are very bright and beautiful. We're now looking at those cerebellar dendrites right down from the top. Lots of color. And here is the spinal cord. These are the neurons that activate muscle fibers that also are expressing in this particular line. I want to talk about these motor neurons that send wires out to the periphery, because that's where we have begun to untangle the wiring diagram first. And here is the wires leaving the spinal cord and going out to the periphery, and each of these wires is a different color. When I first saw these, I said, boy, we have a really good idea here. This is wonderful, and what a great invention. This is going to allow us to track wires over long distances. I was a bit disappointed to discover that computer scientists and engineers had long ago appreciated this trivial idea of painting wires a particular color to trace them from one part of a thinking machine to another part. So, we don't get that much credit for this idea. But it is, we did a biological version of what they had already done. If you trace these wires all the way out to muscle, you can see the synapses these axons make when they contact muscle fibers to make a muscle contract. So this is, again, a three-dimensional data set. I'm spinning it around, and there are muscle fibers that are invisible here running up and down. They're cylinders. Each muscle, you have like a biceps, might have several thousand muscle fibers in it. You can't see the muscle fibers, but you can see that these claspy, pretzel-shaped endings are sort of cylindrical shape. They're sort of hugging invisible cylinders. These are the synapses where a nerve cell from the spinal cord ends, it terminates on a muscle fiber and releases neurotransmitter to cause a muscle fiber to contract. And one thing to notice here is that each of these has one axon, and the axon is color, tells you which neuron it is. So it's pretty clear that these two neuromuscular junctions are from the same nerve cell. Their color is identical, but this is definitely a different neuron and a different neuron and a different neuron and a different neuron. In this way, we could map out all the wiring in muscle. So in muscle, here is a simple cartoon of what a muscle is like. In the adult, you have a bunch of neurons in the spinal cord, and each of them go in and branch to several muscle fibers. Here, I show the branching going to 2, but typically in an adult, an axon might go to 20 or 30 muscle fibers, but each of those muscle fibers is contacted by only one axon. And this is the wiring diagram of an adult muscle. Now, this is just a cartoon, but now with these rainbow techniques, we could actually do this for real. And I'd like to show you what that result is, because there's a painful lesson in looking at this. So here's one of the axons going to a muscle that wiggles the mouse ear. It's called the Interschool Alerus muscle. Most of us don't have them. I have one daughter who clearly has a muscle, but I don't, or at least I can't activate it. But some of you can wiggle your ears, and this is the muscle that does it. Mice are wiggling their ears all the time. So there's one axon in this muscle, and these little endings are those neuromuscular junctions. So there's another axon. You see that other axon kind of covers the same territory. The muscle's kind of a flat sheet, and these two axons go in roughly the same place. And here's a third axon, and here's a fourth axon, and here's a fifth axon, and here's a sixth one. This doesn't go on forever, so don't worry. We're almost done with these. There are 15 altogether. So here we add them all on, one after another. They're kind of an amazing mixture of colors everywhere. There's nothing spatial about this. They don't occupy different parts of the muscle. You see many of these have small arbors, have relatively few branches, and a few have large ones. There's the last one. So that is the connectome, if you will, of the Interschool Alerus. Now, a mouse has 2 ears, and if you look at a mouse straight on, they are perfectly symmetric, just like most human beings. You could just, your left side and your right side looks about the same. So a very simple question is, if we look at this wiring diagram on the other side, is it going to be the same? Is it going to be a mirror symmetry of this one? This is, after all, the same animal. And I can tell you that the left ear and the right ear functionally are identical. So what do you think? Are we going to find a mirror symmetrical wiring diagram on the other side or not? No. Now, let me just add a little more data, since you rushed to this no so quickly. If I did this in an insect or a worm, and I looked at the wiring diagram in one animal, and then I went to another animal and looked at the wiring diagram, they would be identical. They are identical. So are these going to be the same on the left and right side of the same mammal? You still say no. Does anyone say yes? You're wrong. Thank you. Yeah, so that's just a joke there. Every instantiation of this wiring diagram in every single animal was different. Every left was different from every right, and every left was different from every left in every other animal. Every one looked different. You don't find that in insects, which have been around a long time and have kind of a genetic program to build their wiring diagram. You look at a mouse, every one of these is unique. So everyone's unique, but the bad news is it's not even that good. There's lots of things that are suboptimal. Useless loops, premature branching. Let me give you an example of what I mean by not that good. So here, first of all, if you look at the left and right side, just the evidence, these don't look like mirror symmetries of each other. If you look at the biggest one on the left and the biggest one on the right, which have the same function, they're used to really make the muscle twitch a lot, those wiring diagrams look different. Not only do they look different, when you analyze them, they're different. Every one is different. It's not only the biggest one, every single one is different, down to the smallest one. But let's look at the second problem, the fact that they're not that good. Here is one of these wiring diagrams, and all these axons were traced out, but I'm only showing one in red. And I want to focus on this area here, right here. So all of the axons coming into this muscle go this way, except for 2 axons. This blue one, which is a branch of an axon that goes both ways, and in this particular animal, this red axon has decided to go left and not go this way. And I am not so certain I know well enough whether this is a good or a bad idea. So I'm not going to judge and say, who am I to judge, actually, what's a good idea or a bad idea. But so at this moment, I'm still not sure whether it's a good or a bad idea. But up here, it makes a hairpin turn, and it keeps going. And if it goes any further, it'll leave the muscle. Then it makes a hairpin turn and goes this way. All of this is to get this branch here, which innervates that neuromuscular junction down there. There was an easy way to do that, right there. It didn't do that. And it's not that every animal had this. Every animal had its own particular peculiarities. Here's another example. This axon branches here, and one of these branches generates that. That seems reasonable. But the branch doesn't go this way. The branch follows the other branch all the way up here, follow these white arrows, and then peels off, crosses over, and then goes there. That's ridiculous. That's a waste of stuff. And this kind of variability is in every single animal. So where do all these variabilities come from? It's good enough, it works, but it's not perfect. And where it comes from is development. And here's the shocker, I think, about the nervous system. If you think of a baby that doesn't yet know how to walk, and it takes a year to walk, and it takes, as I said, many decades to get tenure, what do you think is happening? You assume that what's happening is you take this brain that has no wiring, and then you become a master by building in these really complicated wiring diagrams. But exactly the opposite is taking place. And this is a very painful thing, and most people don't want to hear this. But when you look at the wiring diagram of a newborn mammal, it is much more complicated than the wiring diagram of an adult. Now, my children will tell you that that makes perfect sense, that I am a simple-minded dinosaur compared to them. But the idea that we start out with wiring diagrams for everything, and then we end up with a small subset of that, is a painful thing to see. So here, for example, is a single axon in it's not an adult, but it's the end of the developmental period of a mouse muscles. At 2 weeks of age, this axon has a subset of branches in a muscle. These gray sites are all the muscle fibers, and this axon occupies maybe 5% of them. And those are those axons I was bringing in one after another. They each have arbors about like this. If you do the same thing at birth and you look at a single axon, you find that it innervates about 85 to 95% of the muscle fibers. Each of them. It's just kind of like crazy. Before birth, there isn't a great deal going on. Everything peaks at birth, and then it's all downhill from there. And this is a very disturbing idea, I think, because it implies you start out as a child with infinite possibilities, and then, thanks to your parents, your siblings, television, those little things you put your thumbs on, I don't know what they're called, cell phones maybe, a vast majority of what you could have been gets trimmed away, and you end up being a rather narrow-minded adult. And I think this being Washington, it is probably worth keeping in mind that there's an important implication here about what happens to adult politicians whose brains now are wired in a particular way. So two politicians might see the same thing, let's say our president, and one of them says, God, I'm really impressed, and the other says, I really am not impressed, and they're looking at exactly the same data. And it's because their wiring now is different. This is a neurological problem. This is, you think I'm kidding, but I think this is really the fundamental problem. If we put all these congressmen together at the age of 5, I think we'd get a lot more done because they'd be more open-minded. But what this implies, perhaps, is that if every axon is doing this, that this neuromuscular junction site should be co-innervated now by lots of different axons. Because if each axon is innervating everything, not only does each axon fan out, but they all must fan into each one of these sites. So let me just show you what that looks like. If we go to one neuromuscular junction, and this is electron microscopy, and we're going to shave through that neuromuscular junction in a block of tissue, and then I'm going to reconstruct back for you. Actually, Juan Carlos Tapia did this painstaking job of reconstructing this. The green is the muscle fiber, and at birth, this is how many axons co-innervate that neuromuscular junction. It's not one, it's many. And I've never been to an orgy, but this is what I imagine an orgy is like. They seem to be perfectly happy to be there. They're all on top of each other. They're all talking to the muscle. They haven't partitioned out any space. They're all over it. One of them is already getting bored or something and has left, but otherwise they're all there. And every muscle fiber we look at birth is like this. And within 2 weeks, all but one of them will be gone. When you're down to only 2 left, at about a week of age, they have already decided they don't want to sleep in the same bed. They segregate to opposite sides of the junction. The red color is the muscle fibers membrane where they release their acetylcholine, and here they are separated from each other. And then we came in and looked at these kind of neuromuscular junctions day after day to see how do you go from 2 to 1. Here you have 2 at postnatal day 11. The yellow axon has a little territory, the blue has a lot. We anesthetized the animal, took this picture, allowed the animal to recover, then came back a day later, found the very same neuromuscular junction a day later, and now you see the yellow has more territory, the blue has given up some territory. A day later, the yellow has taken over more territory, the blue has lost some territory, and by 2 weeks of age, you're down to 1 input, and then that plumps up, and that's the lifelong mate of that target cell. So think of this as almost like 2 suitors competing for lifelong matrimony for the muscle fiber. And once you've got that, that's what you've got the rest of your life. So a decision was made, this is the winner, and that's the one who stays. The winner because matrimony is a good thing. That is, you want to have somebody talking to the muscle fiber to keep that muscle twitching. So what basically, I mean twitching literally in this case. So here's just a cartoon of what's happening in muscle, and I would argue perhaps by analogy in other parts of the brain. You're born with an all-to-all wiring diagram, and then after competition between these nerve cells, each muscle fiber ends up with a different axon. And I could spend a lot of time telling you about the mechanism of this loss, but I think many of you are interested in, well, let's talk about the brain. This is muscle, so I would like to end with that. So more than 90% of the synapses that were formed in development are eliminated in early postnatal life. And could this kind of pruning and simplification occur in the central nervous system as well? And I think to answer that question, we're going to have to look in the cerebral cortex. So this is a picture of the Cajal type Golgi stain, but now with rainbow, where we have every cell a different color. So you might say, well, why don't we just trace out the wires of each of these axons? So if we zoom up on an area like this and try to do that, it's very disappointing because, big surprise, there's too many wires. It's very hard to use rainbow to do this kind of work. If we take subsets of nerve cells in the adult brain, we can see them beautifully. Here is just, for example, the inhibitory neurons that surround pyramidal cell bodies. But that's not what we want. We want to see all the wires. And in order to do this, we had to build a different kind of machine. And so this is a machine that looks a little bit like a movie projector. This looks like a piece of film, and these look like frames of a film. And that is indeed what they are, but it's not a film that we have developed. It is a film that comes from a brain. And I'll explain what this is. This is a conveyor belt that is collecting brain samples from here. So let's see here if I can just get this to switch. So we take a piece of brain, we harden it in plastic, and then we move this chuck up and down against a diamond knife, and we shave off sections that are 30 nanometers thick. 30 nanometers is 1,000th the thickness of a human hair. They're invisible, they're very, very thin. And then we have a conveyor belt that picks one up after another, and we can do about 1,000, and now we're up to 12,000 sections a day. So we can generate a huge amount of brain that used to be a three-dimensional block, and now it's a series of frames on a tape. And we take the tape, we cut it into strips, and we put it on a conductive silicon wafer that's about the size of a CD, about 4 inches across. And we keep doing that until we have a whole library. So we've taken a three-dimensional data set, and we've now turned it into a two-dimensional set of strips. This is what a library might look at like for 10,000 sections. That's about 100 terabytes of data. Here's what one of those wafers looks like. Each of these sections is 30 nanometers in front of the section before it. And if we zoom up on one of those sections, in this case it's about 1 millimeter by 1 millimeter. These are blood vessels here. These are nerve cells. And then this is a very low resolution picture. But in this region here, we took the image at enough resolution to see every synapse. So that's what this image is here. This is an image that goes from, it's 0.4 millimeters by 0.4 millimeters, and it's taken at 4 nanometer resolution. So that's 400 microns by 400 microns at 4 nanometers. That means this image is actually 100,000 pixels by 100,000 pixels. I'll give you a rough idea how big that is. This image here is about 1,000 pixels across, just the way the monitor works. So this actual image is 100 times wider and 100 times taller than this picture. Just imagine a picture that big, 100 times bigger than this and 100 times wider. It would be so big that if I put it here, you'd have to really back up to see it, and then it would look just like this. So there's no point in even trying to show you the whole picture at once. So this is not a megapixel image, this is a 10 gigapixel image. And then we did that 10,300 times. I'm going to show you all those images at once because it shows some of the technical problems here. This is 103 terabytes of data. Here is most of those images. And notice this image and this image. We do this all by automated imaging. We do about a terabyte a day. And when this image was taken, for some reason, the microscope just said, no, I don't want to take a picture right now. And of course, we weren't sitting there all day and all night, so we missed that moment. But it means we have to come back, and here it happened again. And notice here the pictures get darker and darker, and here the pictures get lighter and lighter. This is because these electron microscopes are not designed to generate this kind of throughput. And when I showed this picture to the manufacturer of the microscope, I remember he looked at this and said, look how uniform. It's just beautiful up here. Because we have to go back each time something like this happens. But anyway, they're getting better, but this is a really difficult thing to get a machine that never makes a mistake. And these machines were never designed to generate this kind of data. But at the end of the day, you have a very large data set. You have a 100 terabyte block of brain. It's large in one respect, and it's painfully small in another respect. It is coincidentally the size of a grain of salt, about 400 microns on a side, and that's 100 terabytes. And that took us 3.3 months, 100 days to take that data. So we have to go faster, clearly faster, because if you think about it, if you wanted to do a whole brain at that rate, it would take many, many centuries. And I can't wait that long. And not only me, my graduate students definitely can't wait that long. So we'd like to get this 3 months and 3.3 months, 100 days down to less than 2 days. We'd like to be able to do cubic millimeters in the matter of weeks instead of years. And instead of doing a terabyte a day, we'd like to be doing many terabytes an hour. And I wouldn't have put those numbers there had it not been for the fact that we've been collaborating with a company, Carl Zeiss, who is building us the fastest electron microscope ever. Here's the first prototype. It looks a little, it doesn't look that pretty, and it's gigantic and gigantically expensive. And the reason it's so funny looking is instead of having a single scanning beam, it has 61 different scanning beams in the same microscope. So it's like having 61 microscopes altogether, and that allows us to get over 1 billion pixels per second instead of where we are now at about 20 million pixels per second. There's a normal-sized adult on me. I come up to about here on this machine. It's just a really, really complicated device. The one that is going to be shipped to our lab this summer is svelter, but it's still very tall. Getting it into the building is going to be a big problem, but we've kind of worked out how this is going to be. It's a 61-beam machine because it scans with 61 little beams that are arranged in a hexagon, and that middle beam is what gives that 61. And so that is the kind of data we get. So I want to just show you what the data looks like on this. Bobby Castori, who's been working on the electron microscopy side, has decided to hold his hand here really still so we could zoom up on one of these. He's now holding his breath and going into the electron microscope vacuum. And soon you'll start seeing these blood vessels and the nerve cells and these big dendrites of the apical dendrites of pyramidal cells, mitochondria, myelinated axons, these dark things, and then finally here a synapse on a dendritic spine. So that's one image, but one has to do, basically make a movie of these images. And so here's a movie going through section after section. And again, you see lots of stuff moving. They're not really moving, we're just moving through space. So this cell body is appearing, there's its nucleus as we're slicing through that cell. I know this looks like it was taken in 1903. And that's because of the substrate has a certain amount of contrast in it, and I apologize for that. We've figured out how to get rid of that, but in the earlier movies we didn't. And so you see these things moving from section to section, the myelinated axons, and these big dendrites that are moving. Here's a blood vessel. You see that if you had a child with a coloring book, they could color these things in the same color from section to section. So you end up with a box of brain, and then what you'd like to do is trace out all the material in there. So let me just go to a slightly higher resolution to show you that even between the big things that are moving around are little wires moving around. The brain is just filled with wires. All this stuff are wires, and if you can follow it from one section to another, that is a wire that, in this case, is an axon making a synapse right there, and then it goes on, and it'll make another synapse somewhere. And they just keep doing that. What you'd like to do is give a child a crayon, a lot of colors of crayons, and give them a coloring book where they would go in and color in this object, the same color, section after section. Any child can do that. But to get a machine to do it, because you really have a lot to do here, is harder. And we've been working with engineers that have automated this ability to now color these things in completely automatically. And so once you color them in automatically, then you can follow them from section to section. If you look carefully here, these things occasionally change color, and that's because the algorithms are not perfect yet. They're getting better all the time, but they still have problems. And they don't know what to do with the Golgi apparatus inside cells, so there's all this kind of psychedelic Golgi, but it doesn't really matter to us. We don't care about that. We're really looking at these wires. So after we get the automatic segmentation, we go in and painstakingly check for any errors and correct those errors with computer-assisted tools. I want to show you a little piece of brain where everything has been corrected. So this is a piece of brain where every wire that has been colored in, I can vouch for. They've been checked and rechecked many times. And you're going to notice here there's a green box. It's actually a cylinder that surrounds this green dendrite, and there's this red cylinder that surrounds this red dendrite. These are dendrites that have spines, and the width of the cylinder covers the vast majority of the spines of the red, and the green cylinder, the vast majority of the spines of the green. And so in this area, we have saturated the connectome. That is, there's nothing left to the imagination. Every single cellular process in this part of brain we know about. So let me show you what's in these areas. So there is a red dendrite and a green dendrite, and there they are, and they have lots of spines. But they're not all that's there. There's lots of other dendrites of other cells that have spines that are pushed in against them. And I want to show you all the dendrites together. This arrow just points to that dendritic spine, which is the one that sticks out the furthest. So if we look at all the dendrites, there that is, go back there. This is all the dendrites in that area. There's the red dendrite and the green dendrite, but here's everything else. Now that's just the dendrites. There's also axons in here. In fact, there's 8 times more axons than dendrites, and here's the axons in that same data set. So this and this are sitting next to each other, on top of each other, and that's not all that's there. There are also these supporting cells known as glia, and that's them. That's also in there, and if you put them all together, that's everything. So that's what it is, and let's look at just this red cylinder here at a little higher resolution. And now it's enough resolution you can see the synaptic vesicles that are making synapses, these freckles inside these cells. The yellow is the glia. I want to give you a perspective of this. This area that you're looking at here is, we've made a lot of progress. We know everything about this area, and it is 1/1000th the size of a mouse brain. So we only have to do this 1 billion times more, and we'll have a whole mouse brain. And let me just give you a perspective of that. That cylinder sits in this cortical column of nerve cells, but it's around one dendrite of one of these red pyramidal cells. There's that cylinder right there. So let's see what's in that cylinder. And this is where the talk takes a very depressing turn. There's way too much stuff in there. That is way too much stuff for a human brain to deal with. And remember, we're talking about 1 billionth of a brain, but we've itemized every one of those things. There are 700 axons, there's 83 dendrites, there's 774 different synapses. That's a lot. But now we can do it, and we can just keep going. And I'm just going to end with just give you one picture of the fact that when we began doing this, we realized it's not just random. Actually, which particular axons innervate which particular dendrites is not random at all. They're making very strong choices among the potential dendrites to innervate. So this is that red dendrite, and only shown in this picture are the axons that are synapsing on its dendritic spines. So you'll see the spines sticking out, and there are these vesicle-filled profiles of axons. And all the yellow and green axons are ones that have found this same red dendrite more than once. That is, they find it, they make a synapse, they bump into all these other dendrites, but when they bump into this red dendrite again, they make another synapse. And we find for all these axons, they're quite specific. They don't just randomly innervate cells, they specifically innervate subsets. And this is completely different from what, for example, the Europeans who are trying to model the way the brain works, they're just superimposing axons on dendrites and saying if they overlap, they're going to innervate each other. We find that there are rules about this that are probably related to what's... Fortunately, I'm just about done. So there are rules that determine what stays and what goes, probably related to learning, and that these connections that are strong are due to the fact that axons are making multiple synapses on some target cells and eliminating their connections with others. I just want to end with one final point about the complexity of the brain. I grew up in an age of big ideas. When I began as a neuroscientist, there were lots of really, really provocative ideas out there, and we were unfettered by information. That is, it was an age of big ideas and little data, and now we are in an age of big data and, by consequence, little ideas. So when we had very little information, it was very easy to have a lot of understanding. And I know this seems peculiar, but humans understand things best when they are not belabored and obstructed by reality. And that as information gets bigger and bigger, our ability to understand things gets less and less and less. And this is a challenge for the future of biology, because biology is going from being an age of big ideas and little data to an age of big data. And we don't know how to think about big ideas and big data, but that's ultimately what we'll have to do. Well, thank you very much for your attention.
Take the cue from where the microphones go, so don't take it.
Okay.
So we have time for a few questions. We're going to try and end the question period in about 10 minutes, so we have time for the social hour. When the microphone comes to you, there are going to be some microphone runners, and they're going to give you the microphone. Please stand so that everybody can see you. State your name and tell us whether you're a member of the society or you're a guest, and then ask your question. So our first questioner.
Yeah, I'm James Griffin. I'm a non-member guest. And I wonder if you have a more extended time scale of the complexity peak that occurred at birth and in the week subsequent. How does that go through early childhood and adolescence?
Obviously, I know much more about mice than I know about human. So the mouse muscle reaches its mature peak at postnatal day 12 or 13. From then on in, there's no changes whatsoever. That your motor system at the periphery is hardwired from then on in. It's highly plastic in the first 2 weeks, but by 2 weeks of age, it's over. The cortex is much slower in a mouse, and even at a month and a half, there are signs that it's a younger brain than later. But this kind of detailed analysis, we haven't done this yet. We're just starting right now, actually, to see how these wiring diagrams look in the first couple of weeks of life. It's also interesting to ask, what about the other end of the spectrum of age? There's adult, but then there's old adults, and we certainly get the impression, at least even I get the impression, despite my children thinking I've kind of settled out, that people who are older than me, people in their 90s or past the century mark, are, if they're not demented, are very predictable in their behavior. They really know what's right and wrong. In the sense that they're probably even simpler, perhaps. It's based on wisdom, it's not stupid, although it's a wisdom based on what happened maybe a century ago, so it's a little out of date. But it is an interesting question of what this developmental time scale is in mammals. And humans, we just keep, a lot of us think we keep learning, so maybe we keep doing this very slowly, but we keep learning all the way into advanced age.
Mike Dole, I'm a member. Your cylinder is nothing if not crowded. How do new connections get made through that?
Yeah, so I think most people, the notion is that to learn, you've got to make new connections. I'd like to try to dissuade you of this preoccupation with making things to learn and think of it more like a block that Michelangelo chips away all but David from. So you don't necessarily have to bring new things in. You may just have to exchange. That is, the nerve cells that make strong connections may be driving out other connections from some target cells, and then they just locally sprout to add another synapse. But you don't need things to come from a distance. This has always been the big mystery about learning. How do you learn to associate things you've never associated before? Do you grow axons hundreds of microns or millimeters to find targets? There's just no mechanism for that. Whereas if it's all there to begin with, and you learn by eliminating all but what's there, then it's much more trivial. And I think that's basically how it's happening. Yeah.
Couple of questions, interrelated. I'm Robert Snedden. I'm not a member. I'm a visitor. The related questions, you talk about now we're getting flooded with data. Where do we go with the ideas? Do you still see the standard sort of great ideas, for example, that there is in a sense a canonical cortical column circuit, and we can riff off that? And a somewhat related question, what does all this mean in terms of, for want of a better term, computation, or however you want to term the control that the brain does? Are we still worrying about spikes, or are we going to have to be honest and worry from the bottom all the way up to the top in terms of understanding?
Yeah, so these are both very good questions. So the first question, let me remind myself, the first...
It was about, is there a great idea like...
Oh yeah, canonical circuits. So canonical circuits was a great idea. And I don't want to poo-poo it. I mean, it is, the current state of affairs is that a lot of neuroscience is trying to work out these canonical circuits. And for those of you who are not in with words like this, canonical circuits mean what kinds of cells are interconnected in the brain. A canonical circuit just tells you what kinds of cells are interconnected, but doesn't tell you anything about these quantities of how many of a particular kind and which particular member of a kind make a particular function. And the poverty of the canonical circuit idea is best seen, I think, looking at the cerebellum, where there are 5 cell types, and the canonical circuit has been known since 1950. But nobody knows how the cerebellum works because it's not the canonical circuit. It's which particular member of the canonical circuit is touching each Purkinje cell and which particular parallel fiber is innervating each Purkinje cell. That is where the really interesting stuff is. So it's not that I think canonical circuits are not true, but they're not really what the brain is doing. That's just a parts list of what kinds of parts are innervating each other. If you're a worm, that is all there is a canonical circuit. But in a mammal where you learn, that's not where the learning is. It's not the canonical circuit. It's the subtleties within the canonical circuit. Yeah, and then the second question was, what about spikes? Yeah, so this is not a rendering of activity, it's a rendering of structure. It's a connectionist view. But I would tell you that if the wires are based on experience, they are an instantiation of the firing patterns that generated them. And there is a central dogma we don't understand about how experience engrammatizes itself, that engrammatization algorithm, into wires. But once you understood that loop, then the wiring diagram is in fact a rendering of the experiences, that is the activity, the firing patterns that generated it. So I don't think these things are as different as people often say they are.
Keith Lynch, guest. So if you did get the circuit diagram for a complete mouse, could you then simulate it in a computer? Do you have enough information for that and get normal mouse behavior out of it? I assume there wouldn't be any further learning since it's a fixed circuit diagram, but...
Well, certainly that is the hope, that the wiring diagram is an instantiation of everything the animal has ever learned. So if you had that, you'd be at least at first base, maybe on your way to first base. I'm not sure you'd have a home run, because you don't have the firing patterns of these cells. For example, there are certain types of neurons that fire in bursts, and others that fire rather sporadically, and some that have kind of nonlinear, complicated firing diagrams where they ramp up their firing and slow down. None of that is built into the wiring diagram. So to simulate a brain, you'd have to know things like that. There's this new movie, Inception, which I haven't seen, but I think that is a movie about this kind of thing. So if maybe that would be a place to get some insight. I mean, I think we're very far from simulating brains in silico, but that is certainly the hope, that eventually this will be the substrate upon which one would have an in silico version of brains.
It's 5 after. We're going to take one more question, but you can come up to the stage after the question and answer period, continue your conversation, and get more questions in. So one more question to the back there.
Hassan Abdallah, I am a guest. And the question is about the wiring.
Could you put the speaker closer to your mouth? It's on. You just need it closer to your mouth.
Hello. So, yeah. So it's about closer. Oh, even closer. Okay. About the wiring. That huge wiring and how complex it is to follow each wire. Is there a way, or is there in looking functionally at those neurons or these axons, for example, to look about dopaminergic or serotonergic or catecholamine fibers and see really where they start? How is that connected? Because the value to a big structure, maybe not, although it could be to take more space, it's not necessarily of a critical function or a determining a function as another fiber that may be of a smaller caliber, but carrying a different neurotransmitter message.
Yeah, so the brain, as I've mentioned before, has at the moment an unknown number of different cell types, and many of them differ by, in terms of their biochemistry, what neurotransmitters they release. We've already discovered, just looking at our data, that some of these axons look like dopaminergic axons. They have synaptic boutons, but they don't have close apposition to spines, for example. If this was a large enough data set, we should be able to trace each of these axons back to a cell body. And the cell body shapes of a wide variety of cell types in the brain have already been described. So we would be able to categorize many of these cells biochemically just based on the structure. Biochemically, different cells look different. Inhibitory neurons, for example, have very specific shapes. They're not pyramidal, they don't have spines. So that's one way. Another way of getting biochemical information is to superimpose, excuse me, on top of this wiring diagram, molecules, antibodies, that stain for particular cell types. Because you have over 1,000 sections per cell body, you can probe the same cells 1,000 times with 1,000 different antibodies, which is a lot of antibodies. And then you just have to keep track of what you find in each section. It's kind of a nightmare of information, but it's not impossible. So we're trying to think about that. It's sort of putting proteomics on top of connectomics. But this is going to be essential. It's not enough just to have wires. You've got to know which ones are dopaminergic, serotonergic, GABAergic, glutamatergic.
So let's thank our speaker. And before you go, I want to present you with this plaque, announcement of your talk, signed by all the members of the General Committee.
Thank you very much.
And
It was a pleasure.
thanks so much again. So I have a few housekeeping announcements that won't take long.